At Microbial Discovery Group, we believe microbial research and innovation are the future of solving some of the world's biggest problems. Our exceptional research and production facilities provide the environment for us to do just that. While others might look and act like a grower, MDG is the grower. We have invested decades establishing processes to provide customer assurance in consistent supply and measurable success. Our facility is one of a handful in the world that can do what we do. See how we stand apart from the rest. Imagine you're a customer bringing us a sample. Each sample we receive goes through a detailed testing process before it sees the inside of our fermenter. First, we create cryovials for each organism and begin DNA testing to verify the strain and establish purity of the culture. Then the isolated bacteria are stored in ultra-low freezers. Once we've firmly established the identity of a strain, we simulate the fermenting process on a small scale to discover how we can produce the highest yield. While many companies use a few standard media formulations, we've found it's worth the effort to create a unique formulation for each strain we receive. We test them until we narrow the best 30 to 40 formulations to our favorite 2 or 3. Once complete, we test the fermenting process in smaller 5-liter fermenters to simulate what will happen in the large-scale manufacturing process, developing the methodology in the lab before going to the production floor. Our custom-designed fermentation tanks are modified for higher airflow and more effective mechanical stirring. This is the center of our operation. Like our 5-liter fermenters, our original 5,000-liter tank uses state-of-the-art technology to enable us to monitor and control oxygen levels, jacket temperature, acid base, anti-foam, and everything else needed to make the best possible product. We own 3 additional 10,000-liter fermenters. With the 5,000-liter fermenter, our production capacity is 35,000 liters. To begin the large-scale fermentation process, the tanks are cleaned using automated clean-in-place equipment. We fill a tank with media, sterilize it, and then inoculate it with 1 strain. We grow 1 single strain at a time. That strain then produces nearly 700 quadrillion spores in 36 to 48 hours. As we prepare to harvest the bacteria, we heat shock the fermenter to remove any vegetative cells, leaving only spores. Next, we concentrate the spores using centrifuges to remove 90% of the water. From this, we are left with about 100 gallons of slurry that can either be freeze-dried or spray-dried, depending on which process is least harmful to the spores. We have custom-built our freeze-drying machines specifically for our needs. They create a vacuum under extremely low temperatures to pull the remaining water out of the bacterial cells. Our technology allows us to carefully monitor and control the atmosphere and temperature on each rack within the freeze dryer to optimize the process for the particular sample. When the slurry has become a dry, flaky material, it's ready to be milled into a fine powder of pure bacterial culture. The powder goes into our mobile intermediate bulk containers to blend it up and make it homogeneous. Our spray dryer allows the drying process to be done much faster for spores that can tolerate it, using high heat to spray the liquid out of the bacterial cells. Much like the fermentation process, we perfect the method in the pilot sprayer before taking it to the large-scale equipment. Lastly, we take samples for another round of QC to ensure consistency of the strain before it's packaged into pails. Now that we have individual pure cultures, we can create combinations of multi-strain products in powder or liquid form. Some products have as many as 6 or 7 strains, and we can include surfactants, preservatives, dyes, or fragrances to make them more consumer-ready. The formula will again go through QC to ensure the product is at the formulated bacillus count. Lastly, the products are packaged here in our facility and shipped out to the customer. Our commitment to delivering high quality and safe products is a priority to build and keep trust with our partners. We are highly certified thought leaders, continuously innovating through real science. Redundancy in our equipment is also a huge secret to our success, knowing we can limit machinery downtime to keep promises to our customers. At MDG, we have combined basic research and development and applied research and development with fermentation, scale-up, and large-scale manufacturing. It's a powerful combination that's nearly impossible to replicate. Join us and let's take collaboration to a new level. Microbial Discovery Group. Real science, trusted process, proven success.